After studying this module, you shall be able to understand seminal fluid and its components, understand the structure of spermatozoa, know about the utility of different parts of spermatozoa, and visualize the structure of human prostatic acid phosphatase, semen. Semen, also known as seminal fluid, is an organic fluid that may contain spermatozoa. It is secreted by the gonads or the sexual glands and other sexual organs of male or the hermaphroditic animals and can fertilize female ova. In humans, seminal fluid contains several components besides spermatozoa, proteolytic and other enzymes, as well as fructose are the elements of the seminal fluid which promote the survival of spermatozoa and provide a medium through which they can move or swim. Semen is produced and originates from the seminal vesicle, which is located in the pelvis. The process that results in the discharge of the semen is called as ejaculation. Semen is a massive yellow-white, glary opalescent secretion having a characteristic odor known as the seminal odor. The incandescence of the seminal stain gives a bluish-white color. Semen identification is useful as evidence in sexual assault cases. It can prove that the crime was committed and help identify the criminal. In such investigations, some evidence may help answer certain questions, such as the occurrence of the sexual contact. Affirmative recovery of any component parts of semen from the victim is a solid proof that sexual contact took place. When or in what time frame did the sexual contact occur? This can be assessed by finding the sperm cells at various locations in the body cavities. Can a particular suspect be included or excluded as the potential origin of the semen? This can be achieved by analyzing the genetic profile of the evidence, the suspect and the victim. Several components including the sperm cells, enzymes, sugars, minerals, organic chemicals and vitamins composes semen. 4 glands that contributes their secretion to the seminal fluid are the testes, the seminal vesicle, prostate and the bulbourethral gland. Cowper's gland is another name for the bulbourethral gland. The approximate percentage composition of semen is presented in this table, indicating the contributions from the various glands. It is necessary to understand that sperm cells are produced only in the testes. Therefore, if any person has received vasectomy, then his semen will not include the sperm cells. The other components, however, would be present as usual. The typical ejaculate contains of between 1.5 and 5 ml of semen. We have shown in this table the various glands that are responsible and the approximate percentage of the quantity of semen and their description. In this table, we have shown the glands such as the testes, the seminal vesicle, the prostate, the bulbourethral glands. Seminal vesicle. The seminal vesicle or the seminal glands are a pair of simple tubular glands posterior-inferior to the urinary bladder of some male ant mammals. Seminal vesicles are located within the pelvis. The prostate gland. The function of the prostate is to secrete a slightly alkaline fluid, milky or white in appearance. Next comes the Cowper's gland. Cowper's gland, also known as the bulbourethral gland, is one of the two small exocrine glands in the reproductive system of many male mammals. The bulbourethral gland contributes about 0.1 to 0.2 ml or 5% of the ejaculate. The secretion is a clear fluid that is rich in mucoproteins. The secretion also helps to lubricate the distal urethra. Then comes the testes. The testes are the most essential organ of the male reproductive system. They are the glands where sperm and the testosterone are produced. The testes are contained in the scrotum and are composed of the dense connective tissues containing around 300 internal compartments called the lobules. Each of these lobules contain a number of coiled tiny tubules where the sperms are produced. Testosterone is produced in cells located in between the lobules. The three major important components of semen are produced by the two organs. Testes is the male gonad in the animals, as said earlier. Like the ovaries to which they are homologous, the testes are the components of both the reproductive system and the endocrine system. The primary functions of the testes are to produce the sperms by a process called as the spermatogenesis and to produce the androgens, the primarily testosterone. Both the functions of the testicles are influenced by the gonadotropic hormones produced by the anterior pituitary gland. Luteinizing hormone, or the LH, that is present in the testes results in the testosterone release. The presence of both the testosterone and the follicular stimulating hormone, or the FSH, is needed to support the spermatogenesis process. It has also been shown in animal studies that if testes are exposed to either too high or too low levels of estrogens, such as the estradiol E2, spermatogenesis can be disrupted to such an extent that the animals becomes infertile. The prostate is a compound tubulo-alveolar exocrine gland of the male reproductive system in most mammals. It differs considerably among species anatomically, chemically, and physiologically. The function of the prostate is to secrete a slightly alkaline fluid, milky or white in appearance, that in humans usually constitute roughly about 30% of the volume of the semen, along with the spermatozoa and seminal vesicle fluid. It produces two very important proteins, namely the acid phosphatase enzyme and the prostate specific antigen, or also known as the PSA. As we will see in the later part of this module, the acid phosphatase is a polymorphic enzyme that has been secreted by the other body organs as well as the plants. Therefore, the acid phosphatase secreted by the prostate is known as the prostatic acid phosphatase. In this module, we will study about the biological characteristics of the spermatozoa and the acid phosphatase enzyme. The spermatozoa. A spermatozoa is a motile sperm cell or moving form of the haploid cell that is the male gamete. A spermatozoa joins an ovum to form a zygote. A zygote is a single cell with a complete set of chromosomes that normally develops into an embryo. The sperm cells contribute approximately half of the nuclear genetic information to the diploid offspring, excluding in most of the cases the mitochondrial DNA. In mammals, the sex of the offspring is determined by the sperm cells, spermatozoa bearing a Y chromosome that will lead to a male offspring having X and Y chromosome, while one bearing an X chromosome will lead to a female XX offspring. The sperm cells were first observed by Anton van Leeuwenhoek in the year 1677. A mature human sperm cell has snake-like structure. It has the following parts: head, neck, middle piece and tail. The head. It is spherical in shape, consisting of large nucleus and a dome-shaped acrosome present on the nucleus. Its function is that the nucleus containing the genetic information and half the number of the chromosomes. The acrosome releases a hyaluronidase enzyme which destroys the hyaluronic acid of the ovum and enters into the ovum. The neck, it consists centrioles, which are proximal centriole and the distal centriole, and its function is that the distal centriole gives rise to axial filament of the sperm which runs up to the end of the tail. The middle piece, it is the tubular structure in which the mitochondria are spirally arranged, and its function is that the middle piece is called as the powerhouse of the sperm because it gives energy to the sperm to swim in the female genital tract. Tail, it arises from the middle piece and it is the end part of the sperm. It contains the axial filament. Its function is to help the sperm swim in the female genital tract. It is the main part of the sperm to move. Now let us consider the structure of the sperm cell in little more detail. Nature has smartly evolved multicellular organisms by differentiating cells in accordance to their specific task. The sole motive of a sperm is to transfer the genetic material to the egg cell. Hence, the sperm cells does not include other cellular organelles like the Golgi bodies, ribosomes, etc. These organelles seems unimportant for this task and therefore nature has not included them in the sperms. Similar analogy is also seen in the mature red blood cells or the RBCs where the nucleus is absent so as to incorporate the hemoglobin molecule. So a sperm cell is composed of the head, the middle piece and tail, as I have already discussed. The tip of the sperm cell is known as the acrosomal cap or the acrosomal vesicle, and the acrosomal vesicle contains the hydrolytic enzymes which facilitates the penetration of the sperm into the egg. When a sperm comes in contact with an egg, the contents of the vesicle are released by exocytosis. The head also contains DNA inside the nucleus. The DNA in the sperm cells is densely packed with the help of highly positively charged proteins called the protamines. Then the midpiece of the sperm that contains several mitochondria. Mitochondria is the source of the energy for the flagellum for smooth propagation of the sperm in a fluid environment. And the last part is the tail that called the flagellum. The flagellum is composed of the microtubules. Forensic importance of the spermatozoa identification in the cases that has been disputed or undisputed. So as the number of the cases of rape are increasing day by day, there is a need of detection of the sperm and the semen even if present in a small quantity, no matter how old the stains are. We have to detect these stains precisely. For detection of the sperm and the semen, the most reliable markers for the investigation in cases of rape, sodomy, bestiality, sexual murder, etc. The detection of sperm is an important factor in confirming the sexual assaults. The identification of one or more sperm is a conclusive proof of the presence of semen. In cases of sexual assault, the forensic examination of evidence for seminal stains can actually be considered a two-step process. Firstly, the stain must be located, and secondly, the stain will be examined so as to prove its identity. Possibly, it may even be tested for the blood type of the individual from whom it originated. Furthermore, the forensic scientists can successfully link the seminal material to an individual suspect by the process of DNA typing. In this table, we have shown the approximate duration for which the motile and the non-motile sperms can be present in the body cavities of a female victim. With the help of this table, one can find out the time of the sexual contact. And as you can see in the table, that the motile sperm cells can be present in the vaginal cavity of the victim within 6 to 24 hours of the sexual contact. It is considered that these time limits are maximum and can fluctuate in individual cases depending upon several conditions. And as for the forensic casework, the fundamental part of the semen identification is to look for the sperm cells. However, there are difficulties in the identification of sperms due to several reasons. That is, if a criminal is oligospermic, then his ejaculate may contain a few sperm cells only. Another reason could be the condition of aspermia, where the seminiferous tubules of the testes does not produce sperm at all. If the criminal has undergone vasectomy, which is the surgical procedure for the male sterilization to prevent sperm from entering into the seminal stream or the ejaculate. In addition to these, there may also be other reasons such as the sperm trapped in the clothing not extracted during the textile extracts. Disintegration of the sperms can also take place during the handling of the material, etc. Acid phosphatase. Acid phosphatases or APS are a family of polymorphic enzymes widespread in nature that can be found in many animal and plant species as well. Acid phosphatase is an enzyme secreted by the prostate gland that is present in large amounts in the seminal fluid. It is not unique to the prostate and can be found in other biological fluids as well, including the vaginal secretions. It is therefore considered a presumptive chemical test for the presence of semen, and semen must be confirmed by other means also, that is the sperm detection or the PSA test. Prostatic acid phosphatase or PAP is not a single enzyme but an array of related isoenzymes from a variety of sources. The PAP assay is a well-documented presumptive assay test for the presence of the semen. Acid phosphatase activity is 50 to 1000 times greater in human semen than in any other body fluid. The use of acid phosphatase as a marker for semen is compromised because the vagina is also a source of vaginal acid phosphatase. Since seminal and vaginal acid phosphatase cannot be discriminated, the only approach to differentiating semen in vaginal secretion is by the quantitative analysis such as PSA or the microscopic inspection for sperm. Finding a significantly elevated acid phosphatase level is consistent with the presence of the semen. For example, if semen is present, the acid phosphatase assay is normally very robust. The acid phosphatase is a type of enzyme which is used to free the phosphate groups from the other molecules during the process of digestion. In humans, the enzyme acid phosphatase is secreted by several organs and consequently they are named in according to their place of origin. The table shows the acid phosphatase enzyme produced by the various organs such as liver, spleen, brain, lung, etc. And note that prostatic acid phosphatase was secreted by the prostate gland as well as some other organs. Like acid phosphatase, the prostate specific antigen or the PSA is also found in the seminal fluid. The prostate specific antigen or PSA is a glycoprotein produced in the prostate and secreted into the seminal fluid. PSA is one of the major proteins in seminal fluid with a concentration of 0.2 to 0.3 milligrams per milliliter. Its main function is to liquefy the seminal fluid. This high amount makes PSA a useful marker in forensic science for the detection of even small amounts of seminal fluid. Semen, also known as the seminal fluid, is an organic fluid that may contain a spermatozoa. Semen is a massive yellowish white glary opalescent secretion having a characteristic odor known as the seminal odor. Its identification is useful as evidence in cases of sexual assault. 4 glands that contribute their secretion to the seminal fluid are the testes, the seminal vesicle, prostate and the bulbourethral gland. It produces 2 very important proteins, namely the acid phosphatase enzyme and the prostate specific antigen or the PSA. A spermatozoon is a motile sperm cell or moving form of the haploid cell that is the male gamete. Stop.